Hello and welcome to Biology Tutorial #Bubio. In this video we talk about important terminology with tricks to remember pre-fertilization event, fertilization event, post-fertilization event in sexual reproduction in flowering plants. If you missed the previous NEET practice set on ecology, environment and population topic, then go to the channel and watch or search for my channel only #Bubio. Either tap to the link in description box and watch the previous NEET practice set. Now let's start. Today we discuss on sexual reproduction in flowering plant topics, main 3 events in easy tricks. So let's see. You look at the board. First one, the pre-fertilization events. It's been pre-fertilization events divided by 2 parts. One is male part and another one the female part. First of all, we discuss the male part. First one is microsporangium. What is microsporangium? Normally it's a pollen sac where the pollen grains are developed and mature. After microsporangium or pollen sac, next we see microsporogenesis process. What is microsporogenesis process and what is the result of microsporogenesis? Normally, microsporogenesis process result formation of spore. Either it is called the pollen grains. When pollen grains formation, after this period, again another process will start. The process is microgametogenesis. Microgametogenesis result is formation and development of male gametophyte. This is the main 3 steps of pre-fertilization events on male section. And last of all, we get the main result. What is the main result? 2 male gamete and another 1 tube nucleus. After microgametogenesis process, we get the main result 2 male gamete and 1 tube nucleus. In pollen tube, 2 male gamete and 1 tube nucleus in pollen tube. Look at the diagram. In pollen tube section, first nucleus is TN, it's mean tube nucleus, and to another male gamete. This is the male part. Next, we look for the female section on pre-fertilization event. First phase, megasporangium, it's mean Bubio. We know this on the basis relative position of funiculus, chalaza, micropyle, and the ovules can be classified into mainly 6 types. First is atropous, anatropous, campylotropous, amphitropous, hemianatropous, and cystinotropas. And after this phase, the second step, megasporogenesis. It means formation of megaspore. Then look at 2 process. First one megasporogenesis and second megagametogenesis. It means development of embryo sac. So it is a 2-phase process. First one megasporogenesis. Megasporogenesis in the development of megaspore, embryo sac, while megagametogenesis is development of gamete within megaspore. The development of megaspore takes place from specialized hypodermal cell called archisporial cell. This cell, after various mitotic division, from a megaspore tetrad. It means a cluster of 4 cells. Out of 3 are degenerate, while remaining 1 develops to functional megaspore or embryo sac. So, 1 cell is developed and functional megaspore either converted to embryo sac, and other 3 cells, 1, 2 and 3 cells, degenerate. So, pre-fertilization in female part, we get the main result, embryo sac with 8 nucleate type. Then events, fertilization events. Through the process of pollination, the pollen lands on the stigma. This is a stigma portion, land on the stigma of a female flower. This is the female flower. Pollen grains germinate and tube cell elongate and grows down into style towards the ovule. First of all, pollen tube germinate and elongated and grows down into the ovule. So, start the fertilization process. First of all, pollination, the landing of pollen grains on stigma. Again say pollination. Second, pollen germination and tube. Pollen germination and tube, pollen tube entry into the style. This is a stigma part, this is a style part. The 2 nuclei also move into pollen tube. 2 nuclei also move into pollen tube. Further elongation of pollen tube into style. When pollen tube entry of ovule, the pollen tube can enter in ovule through 3 alternative ways. First one, porogamy, entry through micropyle. Second, mesogamy, entry through integumentary, and chalasogamy, entry through chalasal end. Next, phase, release of sperm cell of male gametes within embryo sac. First fertilization, 1 egg mating of male gamete. This process is called true fertilization. Either it is called syngamy, the result is zygote, either oospore. It's a diploid. Then double fertilization, it means syngamy with triple fusion. This is called the double fertilization. Why? Because DN, it means definitive nucleus, either secondary nucleus, fusion with another male gamete. This is called the triple fusion, and we get the result PEN, primary endosperm nucleus. Why? It's a triploid because 2N fused with the another N. So we get result double fertilization in angiosperm. This is the fertilization event. Next, we discuss the post-fertilization events. Post-fertilization events. In post-fertilization event, first phase development of endosperm. First phase development of endosperm. Endosperm formed as a result of triple fusion. As a result of triple fusion, it's mean endosperm. Primary endosperm nucleus, either it is called pen. Its nature, it is triploid. As a result of triple fusion, a triploid structure called primary endosperm mother cell, either it is called in a short form PEMC. PEMC. What is this? PEMC. It's mean, again say, primary endosperm mother cell is formed that finally produce a mass of nutritive cell called endosperm through mitotic division. On the basis of development, endosperm is of three types. First one, nuclear endosperm. It is the most common type of endosperm. Primary endosperm nucleus divided rapidly without wall formation, hence large number of free nuclei present. Example, maize, rice, etc. Second, cellular endosperm. Every division of endosperm nucleus is followed by cytokinosis. Occurred in about 72 families, like as Datura, Petunia, etc. Datura, Petunia, etc. It's an example of cellular endosperm. And last one, halobrial endosperm. It's occur mostly in monocots. The endosperm is of intermediate types between cellular and nuclear endosperm. This is called the halobrial endosperm. Example, Asphodelus families. Next, the last phase of post-fertilization events is embryogenesis. Embryogenesis. What is embryogenesis? Before going into detail of embryogenesis, we first understand the embryo. What is embryo? Embryo of a plant is a miniature plant tucked into a fetal position in the seed. It is actually one of the earliest stage in the development of a plant, where nutrient provides to the seed enable it to germinate into a plant. It's mean embryogenesis, it's a development of embryo. And the last phase of the embryo, we get the result, the root cap, either root cap and shoot apex. This is the main three events of a flowering plants First one, the pre-fertilization events, second, fertilization events, and third, post-fertilization events. I hope you liked the video. If you did, make sure hit like and subscribe button and press the bell icon for upcoming important notification. And thanks for watching. See you again.